All right, here we go. There. Hopefully this works. I hope everybody can see this. And I apologize again for the seemingly continuous technical kerfuffles we seem to be having. So hopefully everybody can see these bleeping slides, so let's just go with it. If a parasite spends its whole life inside one host, it's monogenous. This earthworm parasite spends all of its life inside an earthworm, except for a brief period when it gets transmitted. These little football-shaped things pass out into the soil and get swallowed by another worm. There are a few parasites that are not only monogenous, they can auto-infect the same individual over and over. This doesn't usually happen. Most of the time, parasites transmit themselves to new individuals, but this is one that can reproduce inside its host's body. This is the one I was talking about that has persisted in the intestines of World War II veterans who fought in the tropical Pacific and then came home and had this roundworm in their guts 65 years after the end of the war. Parasites that spend different life cycle stages in different species are heterogenous. The definitive host is the one where the parasite undergoes sexual reproduction. The other host, or sometimes more than one host, are the intermediate hosts. So in the case of the cow liver fluke, which we'll be talking about a lot coming up, the definitive hosts are livestock, usually cows, sometimes sheep or goats, occasionally humans, but you have to have kind of a bit of bad luck to get it, because that's where the liver flukes, these flatworms, have sex. The eggs hatch into larvae that parasitize snails, and the snails would be the intermediate host, where the parasites reproduce asexually but not sexually. That was review. Just for fun, this can vary even among closely related parasites. This is one you're going to be seeing in an upcoming lab. This is a tapeworm called Hymenelepis diminuta. The definitive host is normally rats. Humans can get it, but as we'll see, it's not that easy to get. You basically have to eat an infected beetle to get this, and most humans don't eat infected beetles. Yeah, not highly recommended. Anyway, the adult lives in the rat intestine. The worm cranks out tons of eggs. The eggs hatch out in the feces, and the eggs get eaten by beetles. So beetles in this case are the intermediate hosts. Within the beetles, the eggs hatch into little larvae, and the larvae develop into an infective stage called a cysticercoid, which you're seeing at the lower left. If a rat eats an infected beetle, the cysticercoid pops out and transforms into a young adult. And it's the circle of life. And, right, anyway. Sorry, I don't know what came over me there. By way of comparison, Hymenelepis diminuta's closest relative is Hymenelepis nana, which is actually monogenous. The adults live in rats. Eggs pass out in the feces. Eggs that get swallowed by rats that are eating food contaminated with another rat's feces. Those eggs hatch into cysticercoids and then mature adults right in the body of the next rat. So even very closely related parasites can sometimes have very different life cycles. Hymenelepis nana has evidently lost the need for an intermediate host. This one, by the way, is a little more common in humans, usually if they're eating food that's had rats pooping on it, which normally you would not want to do, but sometimes it happens if the food supply is not cared for very carefully. There's cases where you can have the same species be definitive and intermediate host. What you're looking at here is some muscle, some meat, muscle fibers, and inside the fibers you have these oval things with little coiled up spirals inside. These are the larvae of a roundworm called trichinella. You get trichinella by eating infected meat. And if you do that, these little larvae pop out and they grow to adulthood inside the gut. So the larvae live in muscle, the adults live in gut, and the hosts for both stages in the wild are animals that scavenge each other. So this thing tends to ping pong back and forth among rats, wild pigs, and bears, any one of which will scavenge the corpse of any others, given the chance. Bears are quite happy to scavenge. Wild hogs will eat just about anything, woo pig, suey, and rats, of course, will scavenge the other two. And so if you had a pig scavenging the corpse of another pig, you could get, the dead pig would have larvae in its meat, and the pig that scavenges the dead pig would get adults growing in it. So in that case, the same species could potentially be an intermediate host and a definitive host. Really, any of those three could be either a definitive host or an intermediate host. Humans, by the way, can get this as well. This is a pretty good reason for cooking your meat, because if you don't, you might not kill these, and human trichinosis is pretty miserable. Fortunately, humans don't normally pass it on, except in rather unfortunate cases of, getting cannibalized or scavenged by wild pigs or something like that. I guess that was a problem in that Hannibal movie, but probably not the worst problem. Anyway, you can have more than two hosts, more than a definitive and an intermediate. There can be a first and second intermediate host. For the record, I'm not going to test you on these genus names that I'm showing you. These are just examples. They're case studies. You don't have to memorize trematodes in the genus Halopicus. Now, some of these genera we are going to get back to, and I am emphatically going to expect you to know Trichinella, for example, because that's a major one for wildlife. It can also be passed around in agricultural settings, and it causes severe human disease. That one I will expect you to know, and we'll get to it towards the end. But you don't have to memorize Halopicus. I just picked this one. It's a flatworm of a group called the trematodes, aka the flukes. And its definitive host happens to be frogs. Eggs pass out of the frog in feces. The eggs hatch into larvae that parasitize snails. A different type of larva bursts out of the snails and parasitizes a copepod. The copepods then get eaten by dragonfly or damselfly larvae, and the dragonfly or damselfly larvae get eaten by frogs. So this is a case where we've actually got three intermediate hosts, plus the definitive host where these worms have sex, and that's the frogs. So life cycles can have more than one intermediate host. You can also have what we call a reservoir host, and this is important not just in parasitology, but in public health and bacteriology as well. It might have been drowned out by the news about the coronavirus, but you might recall that maybe last month, we had a few cases of bubonic plague in the western United States. That's actually fairly common. It usually doesn't break out into mass epidemics the way it did back in the Middle Ages, when the Black Death killed off roughly a third of the population of Europe. But we do have periodic cases of plague in this country, and the reason is that the bacterium that causes it, Yersinia pestis, is prevalent in rodents, including this one, the black rat. And as long as you have rodent populations, which we've always had, you've never truly gotten rid of rats. As long as we have those, rats can be a reservoir for the plague bacterium to jump to humans, which happens, I think, in most years. We'll actually get at least a couple of cases of plague. Although these days we've got treatments and we've got public health monitoring, and it hasn't yet become a true epidemic. But it will probably never completely go away because this very common species is the reservoir host. That's bacteria. If you want a protist example, down here on the bottom, these red things are single-cell protists in the genus Leishmania. They are not an enormous problem in the US, but in many parts of the world, Leishmania causes severe human disease. In the Middle East and Central Asia, it causes a disease called Kala-Azar, which can wreck your spleen, among other things. It's transmitted, by the way, by the bite of infected flies. If you get bitten by a sandfly, you might get this. There's another species of it in South America that can cause very severe skin damage, huge weeping ulcers and things like that. And it's transmitted to humans by the bite of blood-sucking flies again. And the reservoir host in this case are South American possums, like this one. We have, of course, the North American possum. Got some in my yard, in fact. I'm sure you've all seen those. But the North American possum's a recent migrant. There's something like 60 South American marsupial species, some of which are related to our possum, but they're only found in South America. Ours migrated relatively recently, sometime in the past 4 million years, probably more recently than that. So anyway, Leishmania populations can maintain themselves in South American possums, domestic dogs, and South American caviomorphs, which is a major group of rodents. Caviomorphs include guinea pigs, for example. So if you're in South America, as long as you have wild possums and wild caviomorphs running around, they maintain this reservoir population of Leishmania that can be passed to humans if you ever have blood-sucking flies that feed on a possum and then bite a person. Okay, am I making sense there? One means of attempted infection control might be to wipe out the reservoir host. In this case, that would not be practical, and it would also have probably very bad ecological effects. And besides, these South American possums are kind of cute, and it would be a shame to lose them. There might be other cases where that's a realistic means of infection control. But in the case of South American leishmania, probably not. You can also get parasites that will go through a stage called a transport host. The other word for this is a paratenic host. And in the case of a paratenic host, the parasite will enter the host and stay inside it in an inactive state. And usually it does not cause a great deal of harm to its paratenic host. This is not always easy to tell from foracy, like the remoras hitching a ride on sharks or something like that. The paratenic host basically gets the parasite from A to B, but it's not usually severely infected. And in fact, sometimes the paratenic host is optional. The parasite may not even need to find a paratenic host in order to complete its life cycle. There's a protist related to a group called the coccidia. Anybody that wants to be a vet, you're going to hear about coccidians because coccidiosis, there are species of it that will wipe out your chickens. There are other species of it that infect rabbits. There's others that infect dogs and cats. Coccidians are a pretty common cause of things like puppy diarrhea. You ever have puppies that have severe diarrhea? It could be coccidians. On the other hand, they could just be mean. So I'm thinking about a dog we used to have. Never mind. Anyway, so related to coccidians, you have a critter called hepatozoans, and it spends part of its life cycle in ticks. And you've all seen dogs biting themselves trying to get rid of ticks, right? If a dog bites and swallows an infected tick, it can parasitize the dog directly. On the other hand, if the tick parasitizes a rodent or a rabbit or something like that, the parasite can inhabit the rodent or rabbit. Hey, I'm a poet and I didn't know it. The parasite can inhabit the rodent or the rabbit and then pass into the dog when the dog eats the rodent or the rabbit. In that case, the rodent or rabbit would be a paratenic host. The parasite catches a lift in the rodent or the rabbit, but does not do it a great deal of harm. We'll actually see, I hope, an example of this that's pretty common in Arkansas. There's a number of parasitic worms that feed on fish-eating birds. And the adults will live in the bird guts, the eggs will be pooped out, the eggs hatch into larvae that parasitize snails. And when they come out of the snails, they'll form cysts on the outside or sometimes in the muscle of a host fish and just kind of sit there and wait for the fish to get eaten by a fish-eating bird, like a heron or something like that. Sometimes these can cause harm to the fish. There's a thing called white grub disease where they burrow into the fish's liver. Kind of fun to see that. There's another type called black grub where they will form cysts on the fish's skin that look like little black spots. Large numbers can make the fish sick, but small numbers probably don't affect it very much. So the fish in this case would be a paratenic host. The parasite is pretty dormant. It's not trying to reproduce. It's just waiting for that fish to get eaten by a heron or some other kingfisher or some other fish-eating bird. Righty-ho. Oh, this is fun. Sorry about the picture. That's a German Shepherd's kidney that has been basically completely replaced by a giant roundworm called Dioptophyma renali. Yeah, the kidney is basically reduced to a little shell of tissue surrounding an enormous worm. Yes, you. Anybody still want to be a vet? Anyway, it can parasitize your dogs, but in nature, the definitive host would be mink. Right? Relatives of weasels, little mustelids, mink. Eggs pass out in the urine, as you might expect from a worm that takes over your kidney. The eggs hatch into larvae that parasitize aquatic oligochaetes. What's an oligochaete? Earthworms are oligochaetes. It's a subgroup of the segmented worms. And earthworms are one example, but there's lots of very small segmented worms that hang out typically in fresh waters. Tubifex worms are one that you might have heard of because those get sold in freeze-dried form as fish food. So if you've ever kept a tropical aquarium, you might have heard of tubifex worms. And there's a bunch of others, and they're very common in most bodies of fresh water. So if our mink or unfortunate German Shepherd urinates in or near a body of fresh water, the eggs will fall in, hatch into larvae, parasitize these earthworm relatives. The paratenic part is here. If a mink or a dog eats an infected worm, it might be parasitized directly. Now, minks and dogs would not normally try to do this sort of thing, but let's say if they're lapping up pond water, they might accidentally swallow one. So they don't deliberately seek out these worms, but you know, if you're walking your dog in the park and the dog just goes over and starts lapping up the lake, the dog might pick up some of these worms accidentally. But the worms are much more commonly eaten by fish. And if a fish eats an infected worm, the larvae form cysts inside the fish muscles. And this is one shown here in cross-section. All of those circular things that you see are cross-sections through a larval worm. They don't seem to cause much harm to the fish. But if a mink eats the fish, or if a dog eats the fish, maybe your dog goes to the park, drinks some of the lake, and then scavenges a trash fish that somebody left behind or something like that, then that's how the definitive host can be infected. So in this case, you have a parasite that may or may not infect a paratenic host. It's not a necessary part of the parasite's life cycle. Under the right conditions, the parasite can be transmitted without it. And the parasite doesn't seem to do a lot of harm to its paratenic host. Though that's not always the case. That white grub is really something to see. All right. By training, I am an evolutionary biologist. I put the evil in it. I have been teaching evolution in the state of Arkansas for 23 years now, and the lightning hasn't struck yet, so we should be safe here. We don't really have time to go into the model of how new species are formed, but you probably had this elsewhere, and I'll go through it briefly. This is not the only way that new species can form, but it's a common one, and it's a model called allopatric speciation. Again, I will never ask you for the Greek and Latin meanings of words, but if it helps you retain the vocabulary, you can learn them. Patria is your fatherland, right? If you love your patria, you are patriotic. Okay, pater is Latin for father. Patria is your native land, your fatherland. Tomorrow belongs to me. Anyway, Right. Allo is Greek for other, as in allele, or an allele of a gene is another form of it, right? Allopathic medicine is medicine that looks for the causes of disease in other things coming in from outside, like bacteria, as opposed to osteopathic medicine, which has kind of a different theoretical basis. Allopatric speciation means different fatherland. And to start it, you have a population of something, animals, plants, whatever it is, living in a region, right? Living in a common homeland. And then that population gets divided by a barrier that its members cannot easily get across. This could be because the population stays where it is and a physical barrier splits it. Maybe a river changes its course, maybe a mountain pops up, maybe somebody builds a highway right through the territory, maybe two continents rift apart because of plate tectonics, something like that. We call that, by the way, vicariance. Alternately, you could have a population and some members of it get across a barrier that's already there that they don't normally cross and found a new population on the other side of the barrier. This is what happened in the famous example of the Galapagos Islands. You've heard of them? Maybe yes? Okay, you might remember there are a bunch of birds in the Galapagos called the Galapagos finches. They are closely related to finches on the South American mainland. We know the species that they're closest to. It's in a genus called Tiaris. And the Galapagos have never been connected with the South American mainland. They've always had about 600 miles of open ocean. Little birds do not normally fly across 600 miles of ocean. But at some point about 4 million years ago, they did. Some population of grassquits must have migrated from the South American mainland to the Galapagos Islands. Maybe they got blown there in a storm. Maybe they just refused to ask for directions. Who knows? One way or another, they became established on the Galapagos, and given time, they've given rise to new species. Once those populations are split, they can't exchange alleles. So any new allele that appears in one population won't make it to the other. The split populations may be subject to different selective pressures if the environments are different on either side of the barrier. The South American finches that made it to the Galapagos found themselves in a rather different environment, different sources of food, different climate, and all of that would have caused natural selection to start altering the new finch population. And the populations grow increasingly distinct, and it may get to the point where you have new species. And we've got tons of examples of this. If you're really dying to know, I cover a lot of them when I teach evolution, and I believe the other professors do as well. So take Biology 4415 down the road if you haven't already. People in the room, have any of you taken evolution? Okay, so at least 3 of you have heard of this before. Okay, great. Now here's the thing, if parasites depend on their host, you might predict that when a population of the host splits, then the population of parasites should also split. Let's say those South American finches have worms living in their guts. I assume that they do. And then a few of those South American finches get blown off course and end up in the Galapagos Islands. Well, not only is the bird population split, the worm in the bird population is split. And then the worms that are now isolated in the birds on the Galapagos are also subject to selection. They'll be subject to mutation. They'll be subject to a change in allele frequencies called genetic drift. And they'll act on the parasite populations just as they act on the host populations. So as the host is speciating into 2 new species, any parasites on the host should also be speciating into new species. And we call that cospeciation. That means that if you take, I assume you've all heard of cladograms, right? Representations of species common ancestry? Yes?
Yes.
That means that if you take a host cladogram and a cladogram of the parasites that live on the host, and you lay them one on top of the other, they ought to look identical, right? Because every time 2 new species of host arise from a common ancestor, 2 new species of parasite should arise from a common ancestor. So here's a fun example. In red, you have a cladogram of primates. Let me see. Up there at the top, the Catarrhini is African and Asian monkeys, apes, and humans. And we're all of us parasitized by intestinal pinworm in the genus Enterobius. This is one that some of you might have heard of. This is the kind of thing that tends to infect little kids. Why? The adults live in the lower intestine. They crawl out at night and they lay their eggs on the folds of skin surrounding the anus. The little kid's got an itchy butt. He scratches. He doesn't wash his hands. He puts his fingers in his mouth or in some other little kid's mouth. I've had a son in daycare. I know how this goes. Some of you may as well. And the pinworm gets passed on that way. It's a monoxenous parasite. And we get Enterobius formicularis. Our close kin, the chimps and the gorillas, get Enterobius as well. Platyrhini, that's the South American monkeys, get parasites in a closely related genus Trypanoxeurus. And Old World monkeys and New World monkeys are each other's closest kin. And the parasites Enterobius and Trypanoxeurus are each other's closest kin. If you look down lower, Nycticebus is, all of the ones below that are lemurs and their kin. So Nycticebus is the slow loris, which I think was viral for a while because people were taking videos of like petting slow lorises on the belly and they go, yeah, there's a bunch of videos of slow lorises being cute, if you care to look any of them up. Below that, Dobintonia, that's this creepy Madagascar thing called the eye. And below that, we've got various lemurs. And you can see that the cladogram of the parasites, which is in black, almost perfectly mirrors the cladogram of the host, which is in red. Almost every time that a lemur species has divided into 2, the host have divided into 2. The parasites have also divided into 2, and they match each other almost perfectly. Notice I said almost. There's a host switching event if you look down at the base of the branch that's giving rise to Nycticebus, the slow loris. You might be able to see that there's a parasite that infects Nycticebus, and that should have split into two lines, one of which would go to Nycticebus. Well, okay, there's the one for Nycticebus. And then if you look, there's a mismatch around the base of the branch that leads to Daubentonia, the ii. You would expect that their parasites ought to be most closely related to Microcebus and Chirogalius, the dwarf lemur parasites. But in fact, they're not. Their parasites made a jump from the ancestor of the parasites of the slow loris. I may not be explaining this very well, and I don't think you folks in internet land can see me when I'm pointing at the slide, but right here is where they don't match. And that's a case of host switching. It sometimes happens that parasites may end up infecting a host that is not closely related to the host that they're already in. This happens. It hasn't happened a lot in the case of pinworms, but it's happened in other areas. This is all from a paper that came out in 1999, by the way. This is a close-up of, down at the bottom, you have Platyrrhini, that is South American monkeys. They're parasitized by a parasite called Trichinoxyurus. And somehow, some monkey parasite managed to make a jump into these squirrels, where it says Sciurus niger, the black squirrel, Sciurus carolinensis. I think that's our common dog chew toy squirrel. Right, you know the one, that's our common commit suicide by running in front of your truck squirrel. They're parasitized by parasites that are most closely related to South American monkey parasites. And so what must have happened must be that some squirrel came into contact with monkey poop back a couple million years ago or so. We don't need to know exactly how it happened. But the parasites of the monkeys were able to take up residence in the squirrel and colonize a host that in this case is not that closely related to them. And so the ancestor of squirrel parasites came from a monkey, and then a branch of the squirrel parasite tree was able to infect another type of host, flying squirrels, glaucomenes. So in this part of the tree, we could actually see there's evidence for at least two host switching events. Reconstructing when and how those happened is not a simple problem. There's some computation that's involved, and there may be more than one possible scenario, but this does in fact happen. You don't always get parasite cladograms that perfectly match with the host cladograms. Sometimes they are very close. Sometimes they are downright spaghetti. This is one that's clinically potentially important. Malaria is one of the global killers. Last I looked, we had over 400,000 malaria deaths globally every year, and it used to be a lot more than that. This is actually one of the things that the Bill and Melinda Gates Foundation has been spending an awful lot of money on, trying to come up with really effective treatment and prevention of malaria. We'll talk about that later. What I'll point out is that humans don't just get one species of malaria parasite living in our bloodstream and blowing up our spleens. There are 5 species of malaria that cause infection in humans. The big killer is Plasmodium falciparum. There's others. There's Plasmodium vivax, Plasmodium ovale, Plasmodium knowlesi, Plasmodium dohterrii, that all infect people and cause subtly different symptoms and are prevalent in different parts of the world. Plasmodium falciparum is hard to see. It's up at the top right of that picture, and it's just been abbreviated PFA. That infects humans. Humans' closest relatives are chimps, and the closest relative of Plasmodium falciparum, Plasmodium rickenoi, also infects chimps. So in that case, we're parasitized by P. falciparum. Our closest relatives are parasitized by a close relative of P. falciparum. Okay, that much makes sense. But the other 4 human malarias are the product of host switching. We seem to have gotten P. vivax and P. knowlesii not from chimps, but from macaques, small monkeys. And exactly how that happened, I'm not sure. I'm guessing we lived close to a bunch of macaques for a while, and mosquitoes kept transferring parasites between species. And I'm not sure how the details, and it doesn't matter here. But what happened in this case is that we acquired species of parasites that were not present in our closest relatives. We got them by this process of host switching. Okay, oh, good grief, I've gone long. Very last thing. Sometimes parasites will speciate when they're not switching hosts. Human head lice and body lice are different a bit. They can still interbreed, but they usually don't. One of them lives on your head, and one of them lives on your body, and in particular lives in clothing. Presumably not in yours because you all do laundry, but in many human populations you'll get body lice in your clothing and head lice on your head. They split an estimated 72,000 years ago. We've estimated that from looking at their DNA, and very roughly that's about the time when human ancestors started wearing clothes. So the idea is originally these lice just infected your head, but when your distant ancestors decided that wearing woolly mammoth skin was just the thing for the fall season, some of these lice populations took up residence in the clothing. They found a new niche, right? They could bite you in new areas. They were subject to different conditions, and they have not completely speciated because they can still interbreed, but they're on the way to doing it. And the reason is speciation without a host splitting. We just created a new niche on our own bodies when we started wearing clothes. And when we started walking around in mammoth skins going, I'm too sexy for my car, too sexy for my car. Old song, you probably have never heard it. And the lice said, hey, wow, new opportunity. And there we go. Okay, right. I will now check the chat. I will stop recording. Sorry for keeping you folks just a tiny bit long. Thank you for putting up with me, and I swear by the end of the class I will get the Verschlugener technical glitches ironed out. Okay, I'm still presenting, so I'm stopping recording now. Y'all have a good day, and I'll see you soon.